Hi students, this is Lisa Schimmel, Professor of Microbiology at Crafton Hills College, and it's time to talk about the second unknown. Pretty exciting because we're getting close to the end of the semester, and I think you're going to enjoy this project. Most of my students do. Let's pick up the discussion with the lab manual, and on page 227, that's the beginning of the information about the second unknown. Let's talk about how this is going to work. Now, this is going to be your only introduction to the second unknown, so when we come into lab on the day that we're scheduled to begin the project, I'm going to assume that you know what you need to do. So let's talk about that. Okay, page 227, lab manual. The first thing you'll do is you will go up and see Renee, and you will select your unknown from a test tube rack that she'll have with a number of bacterial cultures in it. And then the first thing you want to do once you receive your unknown is head back to your workstation, and on the report form in the lab manual, page 231. All right, I don't know if you're going to be able to see this or not, but it looks like this. Anyway, what you'll do is you'll come back, and in the upper right-hand corner where it says culture number, you're going to write down the number of your unknown. Please, guys, make sure you do that. If you have to come back and ask either Renee or I the number of your unknown, we're going to deduct a point because it really doesn't take a whole lot of effort to just write that number down. All right, then what you'll do is this. Before you do your gram stain, a couple of things you need to do. Number one on page 231 is entitled Cultural Characteristics. So you're going to make some observations of the growth on the slant. You want to address the texture of the culture. For example, is it dry and rough, wrinkled? Is it smooth, moist, shiny? You might want to indicate if there's any pigment being produced, and how much growth is on the slant. Is there a lot of growth? Is there just a little bit of growth? So that's what you'll address in item number one, culture characteristics. Okay, now the next thing you're going to do is, and this is a little different than what we did in the first unknown, is, and this is going to happen before you do your gram stain, you're going to obtain 2 fresh TSA slants. We usually have a basket of them on the side bench where we usually put the materials. And what you're going to do with those is, before you make any inoculations, you're going to label those tubes. Put your name on both of them, put your culture number on both of them, your lab section would be good, and then on one tube write stock culture, and on the other tube write working culture. Then what you're going to do is, using the original tube that you got a little while earlier, you're going to subculture your unknown onto those 2 TSA slants. You can put those, we'll let you know, either in the incubator or depending on what day of the week it is, we may have you incubate those in your lab drawer. So you're going to let those grow up between when you start the project, when you inoculate them, and the next class period. And then those are going to be the organisms that you're going to work with throughout the project. And when you're done doing what you need to do on the first day, you will be returning the original unknown to us, and we will essentially archive it until the end of the project in case there are any issues that may happen to come up. Right, now let me explain the purpose of the stock and the working cultures. Now when you come back next class period, make sure that they grew well. And the stock culture, that's your really your emergency backup. What I mean by that is this. If during the course of the project, either myself or Carol or Renee should say to you, I believe your culture may be contaminated, then you would, and only then, you would go to the stock culture, which should be just hanging out in your lab drawer throughout the project. You'll go back to that stock culture and you'll streak a new working culture from it. Or let's say you dropped your working culture on the floor and the tube broke. That would be another reason that you might want to go into your stock culture. But your stock culture is your emergency backup, and if all goes well with this project, you'll never open that stock culture until the very end when you're just going to loosen the screw cap and boil it down for disposal. Right? So that's the stock culture, emergency backup. Now the working culture, that's the one that you're going to use when you need organism to inoculate the various tests that you'll be performing during the course of this project. And I would highly recommend that once a week, and you don't need to ask permission to do this, we should have a basket of TSA slants on the side bench available for you to use, and if we run out, just let us know, we'll get you more. But anyways, your working culture, you should once a week create a new one from your old working culture, not the stock culture, and let the new working culture grow up. Make sure that you've got sufficient growth on the slant. Once you see that's happened, then you could go ahead and boil down and dispose of your old working culture. Now, the reason we do this is so that you have enough organism to inoculate your test, and also so that the organism is relatively fresh throughout the project. Okay, so stock cultures, emergency backup, working culture, that's the tube that you're going to actually remove microorganism from to inoculate the test that you're going to be performing. Okay, once you have inoculated your stock culture and your working culture, then you're going to do a gram stain. And I will be setting a time limit. I'll make sure that I give you enough time to make the inoculations you need to make and do the gram stain. And then you will obtain one of those gram stain report forms. It's the same form that we used for the first unknown, and you'll fill it in just like we did for the first unknown. We'll need to know your name, your culture number. There's some class information that you need to circle. And then all we want to know at this point is the gram designation of your unknown, your choices being positive or negative, gram positive or gram negative, and the morphology. And your choices are cocci or bacilli, or cocobacilli, if that's the case. And for right now, we're really not interested in the arrangement of the cells. So, I mean, are they in staph formations or strep formations, or are they just scattered around the slide in no particular arrangement at all? And by the way, you'll after you complete your gram stain, you'll take a look again at the report form on page 231, and under item number 2, you can report the results of your gram stain there, and you can also indicate the arrangement of the cells on that report form. Okay, so you do your gram stain, and you will verify the results with the lab tech. The gram stain for this project is going to be worth 10 points. It was worth 5 points for the first unknown, so take your time when you do your gram stain, do it right. And 5 points for the gram designation, and 5 points for the morphology. And from there, we will go ahead and move on. What will happen next is, if you look at page 227, it says use any other staining techniques that apply to your problem. It's highly unlikely that anybody here will need to do any stains other than the gram stain. Oh, and I wanted to mention about the gram stain. Make sure that you review the procedure before you do your gram stain. It's been a number of weeks since we've done one, and I want to also remind you that a very thin and uniform smear when you're doing your smear prep, that's really one of the most important points of the process. You want a nice thin and uniform smear, no thick and thin areas in it, and that's going to set you up to get the best possible results with your gram stain. Okay, I'm on item number 6 in the lab manual, and it says using your lab manual, Berge's manual, which I'll talk about in just a little while, and handouts. I posted some things on Blackboard that we'll be reviewing this morning. Determine what biochemical tests you need. Don't be wasteful. Inoculate only what's needed. And when we get to the point where your flow charts have been approved and you're actually running the tests, you'll only be able to run each test once. Here's what I mean. Sometimes students feel that they're not confident that they got the correct results. We're not going to let you run the tests over and over again. So reread the procedure before you begin, inoculate carefully, use great aseptic technique, and then you should be able to be confident in the results that you get. Okay, number 7 on page 227, this is the point where you will be given a list of unknowns, and we call these the bug lists. And again, I'm not sure if you can see this, but you should have one of these in your hands by this point. And anyways, your unknown is one of the organisms on this page, and I typically use 10 of these a semester, and each of the pages has somewhere between maybe 12 and 14 organisms on it. Your unknown is one of those, and you're going to write a flow chart to show how you would separate every single organism that's on your bug list. More on that in just a few minutes. I do want to add, though, that the flow chart will be due the next class period. Those dates will be on your syllabus, and we'll discuss those. And you will not be allowed to begin any tests other than the ones we do first day of the project until your flow chart has been approved by me. Okay, I just turned to page 228 in the lab manual. Under item number 8, media and reagents. While I am grading your flow charts, the lab tech will talk to you about the process for requesting materials to run your tests. We have a little form that you guys need to fill out, and you'll list on there what media you need. Like, for example, maybe you need tryptone broth, and then you'll be asked to explain what it is that you're trying to accomplish with this test. But we'll talk more about that when we get to that point. Okay, now also throughout the project, you guys will be responsible for boiling down your organisms, your tests, your test tubes, when you're done running your tests, and keeping your area clean. We'd really appreciate it if you would do a good job with that. Okay, let's talk about this this item on page 228 that says here are some additional guidelines. Right, here's how the project breaks down. It's worth 100 points total. The gram stain is worth 10 points. That we'll be doing the very first day of the project. The flow chart is worth a maximum of 50 points. And then depending on whether you have a gram positive unknown or a gram negative unknown, you'll be running either a catalase test or an oxidase test after you do your gram stain. I'll talk just about that in another minute. All right, now, and that's worth 5 points. I'm sorry, you'll verify the results of that. Of course, we have another little form for that purpose. You'll fill this out. You'll indicate if you did either the catalase test or the oxidase test and what your results were. You will also bring this form to the lab tech for verification, and that test is worth 5 points. Okay, under rare and unusual circumstances, it may be necessary to verify the results of one, or maybe, not likely, 2 tests along the way as you're working through this project. The cost to do that is 5 points. Let me explain what I mean. About once a week, you should gather up your flow chart and come and talk either with me or with Mrs. Burns or Renee, and just let us know where you're at. I've done these tests so far, and these were my results. And we'll take a moment and we'll look at your flow chart, and we'll be very non-committal. We'll probably say something helpful like, but occasionally we will see that a student is way off. And we may say something like, you know, I think maybe you could use some help, and I'd like to verify a test result for you. Now, of course, we're not going to tell you which test that is, and there's, you know, absolutely no mandate that you have to accept our help. But what I'm saying is that it might be smart to sacrifice 5 points to try to capture the other 30 points that will be available for you. Oh, and by the way, sorry guys, the remainder of the project, that means successfully navigating through your flow chart and coming up with the right answer. That's worth 35 points. So, I'm sorry, that's what I'm talking about when I'm referring to 5 points to verify a test result. That would be as you're moving through your flow chart. Okay, now let's do a recap of what you need to accomplish during the first day of the project. First of all, you will streak your stock and working cultures. You'll also make some observations about the growth on your slant. You will perform and verify the results of a gram stain. Now, as it says in your lab manual, if you have a gram positive unknown, then the next thing you're going to do is a catalase test, and then you'll verify those results with us. If you have a gram-negative unknown, then the next thing you're going to do is an oxidase test. Same thing, you'll verify those results with the lab tech. And I think we're going to skip the part that involves using OF basal media, so we're going to omit that part. Okay, so you've streaked your stock and working cultures, you've performed your gram stain, you've performed either your catalase test or your oxidase test. What's going to happen now is that is when we will give you your bug list. It has a number on it. Make sure you record that somewhere. I'm not sure which set we're using right now, but as I said earlier, we usually have about 10 of them circulating. And then what you will do is you'll return the original unknown to a test tube rack that we'll have on the side bench. And like I said earlier, we'll archive those. And then you will gather up your things and you'll head off to the library and you will check out a copy of Berge's manual. Now, here's what it looks like. This is Berge's Manual of Determinative Bacteriology, 9th edition. And we've got several copies of this book on reserve in the library. Now, guys, this is the only source of information that's allowed for this project. Unfortunately, Berge's Manual is not online, so you're going to need one of these paper copies. They are on reserve in the library. I believe they're allowing you guys to check them out for 2 hours at a time. And please, if you would, don't write in the books, don't deface the books. What you'll do at this point is you'll be given one of these papers right here, and this is called the Checkerboard Matrix. Now, the column here on the left is where you're going to write down the names of the organisms that were on that bug list that you were given earlier. And then you'll see the names of a number of biochemical tests on the top. What you're going to do is you'll go to the library and you're going to do your best to fill in this checkerboard matrix. Now, some cautions. Number one, no matter how diligent you are, you won't be able to completely fill in the checkerboard matrix. Here's why. Some of the tests on this list are only used for gram-positive organisms. And if you have a gram-negative microorganism, you're just not going to find test results for that one. And just the opposite of what I said. Some of these tests are only used for gram-negative organisms. If you have a gram-positive unknown, you're just not going to find the test results. So you're going to fill it in best you can, being very careful not to make transcription errors. It's quite easy to make a mistake transferring information from Berge's Manual to your checkerboard matrix. So you need to be careful that you don't do that, because otherwise, when you write your flow chart, you're going to have the wrong information and your flow chart isn't going to be correct. That's one of the things that when I'm grading the flow charts that I deduct points for. I'm looking for errors in you reporting test results. All right, so you fill in the checkerboard matrix, and then you at some point need to make a decision. Okay, I think I have enough information to write my flow chart, and then you'll sit down and you'll start writing your flow chart. Okay, I'm going to break for a moment, and then I'll come back and I'll talk more about the checkerboard matrix, what tests are available for you to use, and how to dig that information out of the Berge's Manual. Thanks for watching. Talk to you soon.